Hello everyone, welcome to my YouTube channel. Today we will start with sexual reproduction in flowering plants of PUC second year. Let us go through some basic concepts first, then we'll go in detail. As we all know that flowering plants have been categorized by presence of flowers, fruits and seeds. We know there are four categories, or it's also called as categories: bryophytes, pteridophytes, gymnosperm and angiosperm. The angiosperm is highly developed, which having flower, fruits and seeds. In this chapter we will go through the sexual reproduction that happens in angiosperms, which bearing flower, fruits and seeds. In sexual reproduction there are three steps basically. First is pre-fertilization event, the events which occur before fertilization. Next is fertilization and post-fertilization. In pre-fertilization we will go through the structure of ovum and pollen grain. Ovum is female gametophyte and pollen is male gametophyte. In fertilization we have fusion of male and female gamete, it's already known. Post-fertilization event, the formation of fruit and seed. So this is a three-step included in sexual reproduction. I hope it's clear for everyone. Before going in the structure of pollen grain and ovum, we should know what is the structure of flower. Basic structure of flower. We have already studied about this in first year, PUC first year. We'll just glance once. This is a typical structure of flower. Example I have taken, by seeing you can guess it's a hibiscus flower. This is a stem. This is a receptacle where all the flower parts are placed on. It is receptacle, or also called as thalamus. The thalamus is having sepals which are protective in function. After thalamus we have pistil. Pistil is a female gametophyte. Pistil is a female gametophyte. It's also called as carpel. It is having stigma, style, ovary and ovules. The pollen tube will study in the next sessions. This is a female gametophyte. Next is stamens. Stamens are male gametophyte. Which is having anther and filament. This structure. The structure that include anther and filament is collectively called stamen. The red color which you are seeing is petal, which are attractive part of flower. This is a basic structure of flower we have already seen in PUC first year. Based on these, the structure and presence of pistil, stamen, sepals and petals, there are four folds of flower. You can guess actually. First is calyx, second is corolla, androecium, and fourth is gynoecium. Calyx is whorl of sepals, corolla is whorl of petals, androecium is whorl of stamens, and gynoecium is whorl of pistil or carpel. So in this chapter, that is sexual reproduction in flowering plants, we will go through in detail about androecium and gynoecium. So we'll start with the pre-fertilization event, that is structure of first male gametophyte, that is pollen. Pollen is on the stamen of the flower. This is a stamen of the flower which is having long slender filament, this is filament, and terminal anther, and this proximal end is attached to thalamus or petals of the flower. In this structure we will able to see that the anther is bilobed, that is two lobes are present with long slender filament. I am drawing to understand easily. This is bilobed, which having each thecas. These are called thecas. One lobe having two theca, another lobe having another two theca. Hence it is dithecus. Okay, I hope it's clear for everyone. Anther is bilobed, which is present terminally on the stamen, and each lobe is having two thecas. Hence it is bilobed dithecus. I hope it's clear. And this anther having a longitudinal groove, that is called line of descent, which helps in the spreading of pollen grains after maturation. So this is a basic structure of stamens on the flower. Next, the number of stamen differ from flower to flower, differ from flower and their species. So I hope it's clear. We'll just once again glance. Bilobed structure of anther, which is attached to thalamus or petals. Number of length may be differ from species. When this is seen transversely, there are two thecas in each lobe. Hence they are dithecous. Now we will see what is the structure of young anther. This is a structure shown in NCERT. Microsporangia. Which are present at the corner of each lobe. Hence anther is tetragonal, which having four thecas at corner. Each one is called Microsporangium singular. This is Microsporangia is plural. This Microsporangia is responsible for production of pollen grains, which is a male gamete. Here I have written Microsporangium, which is singular, which forms pollen sacs in the further development to form, which having pollen grains. Now we will see what is the structure of each pollen Microsporangia. This structure is shown below. Now first we will see what is the structure shown in NCERT, then we will go through it. Each Microsporangia are circular in their structure. Here this is one Microsporangia that is circular in structure. Outer layer is circular, which is having four layers. First is epidermis, outer layer. Next is inner endothecium. Next are 2 to 3 layers of cells having middle, they are called middle layers. Next is tapetum, and next is group of cells, which we'll see what are they called as. Okay, this is a structure which is given in NCERT. For in detail understanding, I have drawn here young anther. This is a structure of young anther. The outer layer is epidermis. Epidermis we easily understanding that it's the outer layer which is protection in function. Next is endothecium, which is having, these cells are cellulose thickening. They are thick walls involved in dehiscence of pollen. When pollen grain is matured and it's ready for dehiscence, this endothecium helps in dehiscence. Then 2 to 3 layer of cells, they are called middle layers. These are middle layers. Next is tapetal cells. Tapetum cells are very important as they nourish the growing pollen grains. They nourish the growing pollen grains and also have pollen kit or callase enzyme. Tapetal cells having many functions, which is having pollen kit and callase enzyme in the process of formation of pollen. It also secrete proteins which recognize the compatible of stigma in pollination. It's also having Ubish bodies which thicken the pollen membrane. It's very vast function of tapetal cells is seen. As per syllabus, as per NCERT, it is only given as nourishment of pollen grains. This pollen kit and is for neat. So next, inside tapetal cells, there are group of homogeneous cells, group of collectively formed homogeneous cells called sporogenous tissue. This center of Microsporangia is having homogeneous cells called sporogenous tissue. This sporogenous tissue having group of cells, they are homogeneous. Here what happens is, there we know that each cell having connection which is called as plasmodesmata. In the process of formation of pollen, there is a connection of cells that breaks. These sporogenous cells break the connection to form individual cells. As we see here, I have shown clearly, these cells break from one another and there is a deposition of cellulose. The cellulose deposition takes place on the cells as plasmodesmata breaks. Now, each cell of sporogenous tissue is capable of producing one microspore tetrad. Each cell is capable of producing microspore. Hence, it is called pollen mother cell, as pollen is formed by these cells. Hence, it is called pollen mother cell. Each cell this will undergo the same process which I am explaining now. Will undergo meiosis to form microspore tetrad. As the word itself indicate, these undergo meiosis as 4 cells are formed in one single cell, that is microspore tetrad. Now, after the formation of microspore tetrad, the tapetal release. In this stage, the tapetum release, and each one from the microspore tetrad forms pollen grain, where the nucleus move to the edge of the cell. 2 vacuoles are formed with the nucleus. Hence, it undergoes mitosis division with a large vacuole here, and asymmetrical spindle are formed. To form 2 unequal cells are formed at the end of Microsporogenesis. This formation of microspore from pollen mother cell by meiosis is called Microsporogenesis. So, the unsymmetrical spindle is formed to form two unequal cells. This large one is called vegetative cell and the small one is called generative cell. This is the structure of pollen grain. We'll go through the structure. First, as this process is clear, up to pollen mother cell, the meiosis takes place. 4 cells are formed. The stage is called microspore tetrad. Then each cell from the microspore tetrad to undergo mitosis with the asymmetrical spindle fibers to form a pollen grain which is having two unequally divided cells. One is vegetative cell which is larger and small is called generative cell. Now we will see what is the structure of pollen grain. Up to now we have seen how the pollen grain is formed. Now the structure of pollen grain, that is male gamete. The structure of pollen grain is generally spherical in shape. Which is about 25 to 50 micrometers in diameter. It is prominently having two layers. The hard outer layer is called, exine, which is made up of a chemical called sporopollenin, which is one of most resistant organic material known. There is no enzyme that is known which degrades sporopollenin. It withstand high temperature and strong acids and alkali. So it is the outer covering of pollen grain as it protects from all the environmental condition. Then pollen grain has a prominent inner wall is called intine, which is made up of cellulose and pectin. It is thin and is made up of cellulose and pectin. On the outer layer of pollen grain, there is a prominent aperture. Where sporopollenin is absent. Absence of sporopollenin is seen on the outer layer of pollen grain, where the pore is called germ pore. Hence, exine is the outer layer, intine is the inner layer. There is an absence of sporopollenin deposition at certain point. That pore is called germ pore. Then this intine is continuously present around the pollen grain, which is very thin, made up of cellulose and pectin. We have seen that cytoplasm of pollen grain is surrounded by plasma membrane. We know that plasma membrane, there are two cells present inside. We have seen in the formation of pollen grain, that is microsporogenesis. This is larger vegetative cell. This is smaller generative cell. Okay. The vegetative cell is bigger and has a abundant food reserve and largely irregular shaped nucleus. This nucleus is irregular in shape. There is food reserves that are vacuoles. And the generative cell that floats on in the vegetative cell, which is having small and floats in that cytoplasm of vegetative cell. So the 60% of angiosperms shed their pollen grain in this stage, the above stage which I have drawn. There are 2 cells in the pollen grain, hence it is 2-celled stage. 60% of angiosperms shed their pollen grains at this stage, 2-cell stage. But other percentage of angiosperms which are remaining, they shed their pollen grain in 3-celled stage. Here what happens? The generative cell undergo mitosis. This vegetative cell undergo mitosis to form 2 cells. Hence the pollen grain has 2 cells as vegetative cell and 1 cell in the generative cell. So it is 3-cell stage. So this is a structure of pollen grain. What are the use of pollen grain for human beings? We'll see that. That is, pollen grain are rich in nutrients. There are many vast number of nutrients present in pollen grain. It has become fashion in recent years that pollen tablets are taken as food supplements. In Western countries, large number of pollen products are formed in the form of tablets and syrups that are available in the market. And pollen consumption has been also increased and claimed in the performance of athletes and race horses. So they are fed by pollen tablets or pollen syrups that they increase their capacity. Okay, now even some pollen allergies are also there. Some of the allergies, that severe allergies or bronchital issues in some people, which leading to chronic respiratory disorder. Chronic respiratory disorder we see in some people due to allergic reaction like asthma, bronchitis. It may be mentioned that Parthenium or carrot grass that came into India has contaminated with improved wheat, which has become in the occurrence of and cause pollen allergy. So now we have seen what is the structure of pollen grain. Finally, the pollen is ready for pollination. Now, when pollen grain is ready, it has to find its stigma, which fall on stigma for pollination. This pollination has to occur immediately or it may take time. It depends on the viability of pollen. Viability means how much they survive before they fall on stigma. Some of the pollens are having high viable time or some of having very low viable time. This period in which the pollen grain remain viable is highly variable, and some extent it depends on prevailing temperature and humidity. It may be 30 minutes In rice and wheat, and it is 1 month in Rosaceae family and Solanaceae family. So once the pollen is fall on the stigma, they continue the process of fertilization. The pollen grains can also be stored in liquid nitrogen in minus 196 degrees Celsius for crop breeding. This process is called cryopreservation. So this is the end of the topic male gametophyte. Next we will see female gametophyte in the next coming sessions. I hope it's clear for everyone. Any doubts, add in the comment section. I'll clear them. We'll see in the next class. Thank you.